So yeah, just to introduce our next speaker, we have Carly Grant from the Department of Plant and Microbial Biology in the University of, from Berkeley, University of California. So it's actually quite a normal time of day for you. It's just become, what time is it now?
It's 2:40 now. It's a
2:40.
Lovely afternoon.
Nice, lovely afternoon. And it's nearly midnight here. So we're approaching the end of the day. So you've got to talk about, it's on the inside that counts, exploring organelles within bacteria, which is fascinating because we're always taught there are no organelles inside bacteria in the same way as you'd expect. So I'm going to pop off the screen so that I'm not distracting you or getting in the way, and hand over to you.
All right, thank you.
There we go. We'll just check you can share your screen okay. Perfect. We can see everything. Great.
Excellent. Oopsies. Oh, I forgot to start at the beginning. Sorry. All right. Hi, everyone, and happy International Microorganism Day. As you heard, my name is Carly. I'm a postdoc in Arash Kameli's lab in Berkeley, California. Please feel free to email me if you have any questions, or find me on Twitter. I'd be happy to answer them or just chat about anything. And today I'm really excited to talk to you about a fascinating area of microbiology on the topic of organelles in bacteria and also some archaea. And that may be surprising for some of you, because when we first learn in cell biology about cell plans of organisms, you might learn that prokaryotes, being bacteria and archaea, are these really simple cells, these stacks of enzymes that lack any sort of structure inside of them or organization. And they're often defined by what they lack compared to eukaryotic cells, which contain a nucleus separating their genetic material from the rest of the cell, as well as other lipid-enclosed organelles that are really important for specific functions in the cell, and have really led to life as we know it today. And so while we can see eukaryotic organisms with our naked eye and how different they are, they really only represent a small portion of the tree of life, as compared to the bacteria and archaea that are really abundant and really diverse and are invisible to our naked eye. And when we start to take a closer look at some of these bacteria and archaea, you'll find that they don't all look like the textbook example of this sack of enzymes. And in fact, some of them also have these specialized, by definition, organelles that carry out specific functions and are really integral for these cells to survive in a particular niche. And here are just a handful of my favorite examples of organelles in bacteria and archaea. And each of these organisms has a different organelle that's really, really important for their lifestyle. For example, cyanobacteria, such as Synocystis, shown here, has thylakoid membranes that house the protein complexes that are essential for the light harvesting reactions of photosynthesis. In addition, these bacteria have carboxysomes, which aren't a lipid-enclosed membrane like other organelles by definition, but they're instead made up of thousands of proteins. And inside this organelle is the important site of carbon dioxide fixation, and is really essential for these organisms to survive. In addition, there are really large organelles that have been found, such as the anammoxysome in some anammox bacteria. These organelles take up about 60% of the cell volume. That's huge. And they contain the important anammox metabolism. And this metabolism creates toxic intermediates, and is perhaps why this organelle is so important for the cell. In addition, this anammoxysome contains tubules of unknown function, as well as iron granules. And most unique about the anammoxysome, or another unique feature, is that in most bacteria and archaea, the ATP generation and the electron transport chain is located at the cellular membrane. However, with the anamoxisome, these processes are carried out at the anamoxisome membrane. Thus, it is maybe more analogous to the mitochondria in eukaryotic cells. And then there's an example of Archaea. So Ignicoccus hospitalis, also are named after a hospitable fire sphere because it is the host of a parasitic nanoarchaea. It's a hypothermophile found in hydrothermal vents north of Ireland at temps around 90 degrees Celsius. It is really unique, not just as being the host for a nanoarchaeon, but because it separates the genetic material from energy conservation, and thus could be a really cool example and great example model system for studying how proteins and membrane trafficking occurs outside of the eukaryotic domain of life. And finally, there are totally unknown bacteria. All they are an observation under a microscope. So here we have Ovobacter propellens, who has the cytoplasm just really concentrated in the periphery of the cell that surrounds a really large vacuole with inclusions. You'll also find in this cell box-like organelles with stacked membranes, and there's no known function for this, again, because this bacteria has only been observed. They can't grow it in the lab. In addition, this bacterium has 400 flagella per cell, and they're all found in a little or a large cavity on one side of the cell. And this bacterium is one of the fastest. It moves at 1 millimeter per second. That would be proportional to humans swimming 300 meters per second and is 100 times faster than the fastest human. And in the Comeli lab, our favorite organelle, or the main studied organelle in our lab, is that found in Magnetospherillum. And these are called magnetosomes. So they're these lipid-enclosed organelles that contain a crystal of magnetite. And they are aligned in a chain in the cell, such that the entire cell will move with the magnetic field. And it's thought that these magnetosomes are really important for the bacteria to find just the right amount of oxygen, because it's a microaerophile. It wants not too much or too little oxygen. So it's thought that it uses the magnetic field to align and search for this perfect area found in the oxic-anoxic transition zone. And we've been studying this bacteria for a long time, both the Comeli lab and also many others. And a lot has been learned about the specific proteins that are important for creating this lipid vesicle, which then is the site of magnetite biomineralization. However, there are many different and diverse magnetotactic bacteria, or these bacteria that make magnetosomes, that have been found. And these magnetosomes are beautiful. There are so many different sizes and shapes, and they're found in so many different diverse bacteria, including multicellular bacteria. And so in our lab, we're also interested in understanding how some of these other magnetite or even greigite, which is an iron sulfur crystal, how they can be formed. And for that, we turn to Desulfovibrio magneticus. And Desulfovibrio magneticus, unlike the model Magnetospherillum species, makes these tooth-like crystals. In addition, it is a strict anaerobe. And so perhaps the hypothesis for the function of magnetosomes is a little more complicated for this bacterium. And so when we try to figure out what magnetosomes are doing and understand how they're forming, we want to first ask just what are the steps of magnetite biomineralization? And this is pretty easy to do. You don't need to do genetics or anything. You simply grow the bacteria without any iron, and without any iron, they won't make the magnetite crystal, which contains iron and oxygen. And then when you spike in iron, you want to see this intermediate phase. So do the crystals form in a chain in the cell, each growing at the same time, or whatever. However, when Dr. Megan Byrne, a former postdoc in the lab, performed these experiments, she found this wild intermediate phase where the cells, within 15 minutes of adding iron, formed filled with these electron-dense inclusions, or granules. And over time, they sort of clustered up, got bigger, and then eventually the cells would make magnetosomes. So she thought, well, are these magnetosome precursors? Because she found that these granules that filled up in the cells were amorphous, unlike the magnetite, that's crystalline. They were bound by a lipid membrane, or just a membrane, and they contained iron, oxygen, and phosphorus. Importantly, both Meg and another former postdoc, Lila Ron Lee, found that they're likely not magnetosome precursors. And this is because the iron from these granules didn't end up in the magnetite crystals eventually. Also, they weren't sufficient for making the magnetosomes. And finally, the genes required for magnetosome formation in D. sulfovibrio magneticus were not essential for the granules that formed. And so when I joined Arash's lab as a graduate student, I was really interested in understanding what these granules were, which we now call ferrosomes. So what I did is I made cells that formed the ferrosomes. I separated them from the whole cell, so I broke open the cells, removed the ferrosomes, and identified the proteins that were stuck to the ferrosomes. And with this, I found a small operon of 3 genes that were highly enriched with my ferrosomes. One of these, of the 3 genes, only one encodes a protein with a functional annotation, and that is a membrane transporting P-type ATPase. And P-type ATPases are found in all domains of life. They are, some are essential, and some, such as these P1B transporters, transport heavy metals or just metals across a membrane, which is pretty cool because we found that ferrosomes have iron in them, so maybe this is a transporter that gets iron into ferrosomes. So my work showed that, or I guess next I wanted to find what other bacteria could make ferrosomes. So I looked at who has the genes for the ferrosomes, and I found a large group of bacteria and archaea that have the potential to make ferrosomes. And I found through this work that many different bacteria that are highly diverse can make ferrosomes. I've deleted the genes and shown that they're essential for ferrosomes. And I invite you to check out our preprint for more information on this. So I feel like I've gone very fast, but hopefully what I've shown you today is that bacteria and archaea are not all like this simple textbook example. They're highly, highly complex depending on where they live and what their lifestyle is. And rather than just calling them these simple stacks of enzymes and just simple cells, I think a lot of textbooks need to change that and say that their complexity is beyond the scope of the textbook, because they're not all E. coli and they're not all so simple. And with that, I would like to thank you all for joining me today in celebration of microorganisms, and thank you to the organizers and Joe and also Pedro for inviting me and sharing the date with me. I didn't know about International Microorganism Day before. And then I also want to thank or shout out to Small Things Considered and also This Week in Microbiology as some real inspirational sources. And with that, I would love to take any questions. Oh, good, I didn't go too fast.
Yeah, well, no, we still have a few minutes.
Perfect.
So I have a question, because I actually, at the beginning of the day, had a talk with Pedro. So is it through magnetotactic microorganisms that you know each other in terms of the research that you've done?
Yes.
Yeah, because I mean, I just can't get my head around the fact that there are these tiny magnets inside microbes and they do all these amazing things.
Yeah.
But yeah, so you showed us an what's it called, an organelle, where most of the bacterium was taken up by the organelle.
Mm-hmm.
So does that mean it has to perform all of its other life functions in the tiny space around the large organelle?
I think so. Mostly there are unknowns with that, I would say. In the tiny space around the organelle, they think that carbon fixation is happening, as well as presumably DNA replication, transcription, translation. So yeah, you would expect it all to be packed in there. But because most of those organisms aren't, or none of them are in pure culture, and none of them have a genetic system, it's really mostly unknown, and they've made a lot of really amazing advances just without genetics, which is pretty awesome.
Okay. And then people are trying to apply genetic methods now to kind of elucidate this a bit more.
Say that again?
Are people now trying to use more genetic and genomic methods to explore the organelles?
I think in only a handful of bacteria, we have the tools for genetics, which is a big holdback. But
okay,
I would think that they're working on getting to genetics, but I'm not sure.
Okay. And yeah, my final question then was, you know how so in eukaryotic cells, things like mitochondria might have their own separate pieces of DNA.
Mm-hmm.
Is this the case with bacterial organelles? Are they more likely to have, you know, other pieces of DNA inside them, or what kind of examples are there out there?
Not that I know of. That's a good point. In most of these bacteria that I showed you, I noted where the DNA was, and that's because the researchers specifically found that the DNA was located in the cytoplasm and the organelles were separate. And with the case of mitochondria, for example, that's probably an ancient bacterium, right? So that's why it has DNA. That's, you know, a relic of bacteria.
Yeah, okay. So these didn't form in that same way, so we might not expect.
Yeah, probably not. Maybe some other organelles in eukaryotes have formed in similar ways, like the photosynthetic membranes could be, I guess, you know, an ancient cyanobacterium, actually, that has its own DNA too. But yeah.
Cool. Well, thank you so much. We've got some more speakers coming in for the final session. But thank you for teaching me some more new things. I've learned so much today. And yeah, enjoy the rest of your day in California. And been, very happy to have you on board. And thank you for your talk.
Thanks. Enjoy your night.
We will. Thank you.